Hello, bachcho. Let's see the question. So the question says, which structure is involved in genetic engineering? So we need to identify out of the three given options. So we need to identify out of the four given options, which is involved in genetic engineering. So the key concept in this question is to understand what is genetic engineering. So genetic engineering is a very important technique in which we can modify gene or alter a particular DNA sequence of a given organism. So genetic engineering is also known as gene modification, or it can be also said as gene manipulation, because here we can manipulate or change the gene of our interest according to our own needs. So as the name suggests, we can engineer gene according to our own interest. So now if we go through the basic steps of genetic engineering, then the first step involved is the isolation of the gene of our interest. So the gene that we want to add into some other organism first need to be isolated from that organism. Then this gene of interest, which is isolated, needs to be inserted into a vector so that this vector can then transmit the gene of our interest into the host organism. So the host organism is now a recombinant because along with its own DNA, it has a foreign DNA, which is the gene of our own interest. So in this way, we can modify the gene of an organism depending upon our own needs. Now, there are a few basic requirements for genetic engineering. So there are certain requirements that we need for conducting a genetic engineering process. So the first one are enzymes. Now there are several enzymes which are required for genetic engineering. A few of them involve restriction endonucleases. So restriction endonucleases are utilized for cleaving and separation of the gene of our interest from the uh, organism's genome, as well as the removal of a segment of the genome from the vector. So restriction endonucleases are used for cleaving of DNA. Along with that, enzymes like DNA ligase is utilized for insertion or the attachment of the gene of our interest into the vector. And the second most important requirement for genetic engineering is a vector. Now, there are several types of vectors or cloning vectors, particularly, which are utilized. One of the most common vectors which is used is the bacterial plasmid. So, the plasmid, which is the extra chromosomal DNA present in bacteria, can be isolated and the gene of interest can be added to this vector. So, plasmid is a very common vector. Other vectors include cosmid, then we have phage, bacteriophages. So phages are also used as vectors, and then we have phagemid. So these are the few vectors that are utilized as vectors. Now these are the requirements of genetic engineering. So now if we come back to our options, then firstly, plastid is a organelle which is present in plants. So in plant cells, the plastids are an organelle which has the chloroplast present in it, which is responsible for the process of photosynthesis. The second option is plasmid. So plasmid is a vector which is utilized in genetic engineering. Then the third says codon. So codon is the triplet nucleotide pair in DNA which is recognized or which plays an important role during translation. So these are the three options which are given. So if we come back to our question, then the question says which structure is involved in genetic engineering. So plastid, since it is an organelle in plant cells, it is not involved in genetic engineering. Second is plasmid. So plasmid is involved in genetic engineering because it is used as a cloning vector. The third is codon. So again, codon is the triplet nucleotide base pairs which is recognized or which is plays an important role during translation. So again, this is not the answer. And since we have an answer, therefore none option will also be not correct. So our final answer for this question is option 2, that is plasmid. It is a structure which is involved in genetic engineering. Hope you understood this well. Best of luck. Thank you.